So I'm Brenna Bray. I'm the founder and director and CEO of the Nourished Research Foundation. I am also an associate professor at the National University of Natural Medicine in Portland, Oregon, in their Clinical Research Masters of Science program. I am core faculty in the Building Research Across Interdisciplinary Gaps program, which is a joint collaboration between NUNM, the National University of Natural Medicine, the University of Washington, and the National Institute of Health, specifically the NCCIH, the National Center for Complementary and Integrative Health. Here's the logo for the Bridge program. I think that ends this year. And then I'm also an adjunct professor at Naropa University in their graduate program, specifically in their clinical mental health counseling master's program, and in the Buddhist informed contemplative counseling cohort, it's a mouthful, as well as in the psychology department, where I primarily teach research, teach some other classes, and am very lucky to be able to be developing a course on the science and neuroscience of psychedelics. So as I mentioned, I did my PhD work on the impact of stress hormones, so this would be cortisol in humans, corticosterone in rodents, on the brain. And a lot of my students, particularly at Naropa, my clinical mental health counseling students, but also my undergraduate students, are really curious about how stress kind of can prime or cause these pathways of mental unwellness. They're really interested in kind of helping support people in healing from that. So they're interested in the mechanisms. And so I wanted to give a little presentation on that. And I kind of had one queued up. And there's a little bit of a theme for today of psychedelic research and psychedelic science. This is something that we see very rapidly emerging both in research and in clinical use. We see, ketamine being used and very widely adopted, psilocybin being deregulated in Colorado and Oregon, actually chronologically Oregon and Colorado, and at 1 to 3 this afternoon Mountain Time, we'll have on Nikos Givasky of Althea, Andrew Novick, who's an MD PhD at CU Anschutz, and then Peyton Follestad, who's a student at NUNM, and they're going to be kind of sharing some research and perspectives on some different psychedelic medicines. So I wanted to weave in that psychedelic component here. I'd also planned to do a lecture on storytelling in science, which is, something that I was taught by many of my mentors and professors in graduate school, but particularly Cliff Summers. And I think can be very helpful for those of us who are in science and kind of want to build up our communication skills. So I kind of rolled that all into one. I have a little bit of an intro lesson that I developed for my Science of Psychedelics course. It's about 30 minutes. It gives a nice little primer to the brain. So I'm going to start by playing that, which is a little bit of a cheat. But I think it's better to play the recorded version than for me to kind of go through it. So I'm going to switch over. I'm going to play that. This will also give me a little bit of a opportunity to give a shout out to our YouTube page. You will notice there's a few times where I say in this class, in this lecture, that's kind of what's going on. And then I'm going to follow that by, looking a little bit more in depth on the impacts of stress in the brain. So let me just kind of quick switch over here. This might take me a minute. Apologies to kind of close. Okay, here we go. End show. So I am on YouTube here, just a little shout out, the Nourished YouTube channel. I'll kind of show after that, but I'm just going to play this video. So you might be starting to feel a little bit intimidated and start to think, well, I didn't sign up for a neurobiology class. I signed up for a Science of Psychedelics class. And so we're going to start with a little lecture that is kind of designed just to help keep things pretty simple, pretty palatable. And I'm going to tell you about how I was taught to tell stories in science. And I'm going to do this using DreamWorks film Trolls. So Trolls, if for those of you who have watched it, if you haven't, this lesson might be a little rough going, but hang in there. Let me know. Please give me feedback on how it is if you haven't seen it. Trolls starts out as a story. And parallel to that, as researchers, as scientists, a lot of our career and what we do really depends on our ability to be good communicators and good storytellers. We need to be able to take these findings, this research, this work that we are so engrossed with and share it with others and help them understand why we think it's so interesting and why they should care. So the way that I usually do that in science is just the same, the way that I was taught to do it is the same way that we see this done in storytelling through the ages, right? And we're going to use trolls as an example. So we start with the landscape. For those of you who have seen trolls, it starts once upon a time in a happy forest, in the happiest tree, lived the happiest creatures the world has ever known, the trolls. They loved nothing more than to sing and dance and hug, dance and hug and sing and dance and sing and hug and dance and hug and dance and sing and blah, 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 right? That gets really fast. So in this introduction, what they're doing is they're providing us an introduction to the landscape that this story is going to take place in the happy forest, in the happiest of trees, right? And then they're also giving us a little bit of an introduction into some of the main characters, and they're focusing more on the protagonists, the good guys, right? And so there's the trolls. They're the happiest creatures that the world has ever known. They love nothing more than to sing and dance. So they're saying, here's the trolls. Here's a little bit about them. We're going to do this in this module as well. We're going to provide you with just a little introduction of the natural landscape of the brain, which is the landscape that most psychedelic interventions occur in and take place in, although they do also take place in other parts of the body and metaphysical realms as well. But we're going to focus on the brain as the natural landscape here. And we're going to introduce some of the neuroanatomy, so different brain regions that are kind of more key players in psychedelic actions. And then we're also going to focus on some of the different neurotransmitter systems that are kind of our protagonists in the story of the science of psychedelics. So these are the main characters, if you will. So storytelling in science, we start by introducing the natural state of things, right? So in trolls, there's the natural landscape. It's the happy forest, the happy brain would be the equivalent in neuroanatomy. And then the key characters. So the trolls were in the brain, the neurotransmitters, and the brain regions. Then we introduce the potential for conflict, right? All good stories have conflict. They have intrigue. There's an antagonist. There's a potential for the whole natural state to become disrupted, right? And so in Trolls, that's the Bergens. And the movie Trolls goes on, but then one day the trolls were discovered by a Bergen. The Bergens didn't know how to sing or dance or even hug. They were the most miserable creatures in all the land. And once they saw how happy the trolls were, they wanted some of that happiness for themselves. So the Bergens are our antagonists. They are the potential to disrupt the system. And so there's this kind of introduction of threat. And so what does that look like in the brain? In the brain, when we're exposed to stressors, the body initiates this response. So here we're seeing a spider that might be the equivalent of a Bergen, right? It has a potential to disrupt the system. If we get bitten by it, it's poisonous. And we actually have some of our own adaptations in the brain that respond to that, right? So when we see a spider, we take that sensory information in with our eyes, and it kind of goes into our brain, but we'll talk a little bit more about that. Here, we're just focusing on there's a potential for the system to be disrupted. Now, in the movie, the trolls get captured, but resilience presides. King Peppy leads the trolls to escape. So the trolls have ways to keep the Bergens at bay or to escape them, right? So King Peppy leads their escape, demonstrating resilience and resourcefulness. Similarly, the brain has its own built-in systems to regulate these threats and respond to them appropriately. So in the brain, we see that spider, we take that stimuli in with our eyes, that goes into a part of the thalamus, the visual thalamus, then back to the visual cortex, and then to a part of the brain called the amygdala. Now, the amygdala is really associated with storing memories of threatening or aversive stimuli and experiences. So how do we know that a spider is bad for us? Part of that involves our amygdala. It rings alarm bells in our brain. When the amygdala gets activated, a few things happen. It kicks off this stress response. So it signals to a brain region called the hypothalamus. This here is the human brain we're looking at here. That might be hard to tell. So it activates the hypothalamus, which secretes these hormones, CRF, and that activates the pituitary, which is also in the brain. The pituitary secretes more hormones, ACTH, which are glucocorticoids. So they're kind of stress hormones on their own. But the big thing that ACTH does is that it stimulates the adrenal glands. Now, the adrenal glands are in the body. They sit on top of our kidneys bilaterally. One really cool thing about hormones like CRF, ACTH, as well as cortisol, which you're kind of seeing here, is they're lipophilic. Which is a fancy word. They're steroids, they're lipophilic, which just means that they can cross barriers. So for example, CRF can be secreted in the hypothalamus, but it can cross the blood-brain barrier to act in the body. ACTH secreted by the pituitary can go off into the blood and again, get all the way down to the adrenals to act on the adrenal cortex. When the adrenal cortex is stimulated by ACTH, it secretes cortisol, which is our primary stress hormone. It's a glucocorticoid. It's also a steroid and lipophilic, which means that when it's secreted into the blood, it has all of these effects that go throughout the entire body, but it can also cross the blood-brain barrier to act in the brain as neurotransmitters, which it does. Now, there are a few regions in the brain that are particularly sensitive to stress hormones like cortisol. They have a really high expression of receptors. Two regions that we'll focus on right now are the hippocampus and the amygdala. The amygdala, you remember, we already talked about, that's the one that responds to threatening stimuli. When glucocorticoids like cortisol hit the amygdala, it just activates and turns on this system, it ramps it up. So it causes us to secrete more stress hormones, as well as the adrenals also will secrete adrenaline and noradrenaline. So these are going to kind of kick in our fight, flight, or freeze response. They're going to help us respond to this threat. Eventually, as enough cortisol is secreted into the blood system, it's going to act on the hippocampus. Now, the hippocampus is involved in learning, memory. That's kind of the dorsal or the like back upper side of the hippocampus. It's also connected to all these other brain regions. It's involved in emotional responses to stress. But one really cool thing that it does is it evokes negative feedback onto this whole system. We call this system the HPA axis, stands for hypothalamus pituitary adrenal, the HPA axis that regulates neuroendocrine, so just brain, neuroendocrine hormone, so hormones that can act in the brain, stress responses. And when cortisol acts on receptors in the hippocampus, it kind of turns that system off. And it's a really self-preserving mechanism. You know, we can't have the system staying on. We'll get overexposure to cortisol. That's not a good thing for tissues. I like to think of the hippocampus as kind of the rheostat or the thermostat for stress in the brain. So if you have a air conditioning in your house or a thermostat, you set it for a certain temperature, right? And then once the house reaches that temperature, it turns off. That's a negative feedback loop. Hippocampus does the same thing. So you see a threat, your amygdala responds, that turns on the stress system. And then when there's enough stress in the body, the hippocampus turns the system off. So that's kind of our stress system in a nutshell. That's one of our main resilient safety and more, I would say, emergency mechanisms that we have to save us when we get into trouble. So here's the hippocampus turning that system off. And then again, with the trolls, they have this escape mechanism, right? So King Peppy led the trolls to safety, where they then kind of remained hidden from the Bergens so that they could be safe. And resilience presides thanks to our stress response and our own innate resilience. But those of you who have watched Trolls know that this is not where the story ends. Okay, so we've covered the natural state of things, the happy forest, the happy brain. We've covered the potential for conflict, the risk for Bergens, threat aversion, and we've even covered some of the ability to overcome conflict and maintain harmony. So the trolls can escape and stay hidden. We have our neuroendocrine stress system, our HPA axis, and these mechanisms that can help us respond to aversive stimuli, threats, and stress. But the story doesn't end there. So in Trolls, that's all just takes place in the first 5 to 15 minutes, right? So what happens when this balance is lost, which we know it inevitably will be? In the movie Trolls, harmony is eventually lost. One fateful day, the Trolls village is discovered and several trolls are captured by the Bergens. So the Bergens take over. This is kind of the equivalent of what can happen when stress takes hold in the body. So here we go. Okay, they're captured. Okay, so this is kind of the equivalent of what can happen when stress takes hold in the body. Now, stress operates on kind of a Goldilocks or an inverted U-shaped curve, which you're seeing right here. So too little stress, and we're not going to have enough motivation to really do anything. We're going to be inactive. We're going to be, too laid back. We're not going to have motivation to get things done. Let me take a step back. Most people think of stress as being a bad thing, right? It feels uncomfortable. Nobody likes to get stressed out. I do have to add the caveat here, stress actually does play a really important role in our lives, right? So if we have too little stress, as you can see here, we don't have really motivation to do much of anything. That stress can be a really good thing. When we start to get into optimal levels of stress, that's when we really see performance peak. But if we get too much stress, we'll start to get exhaustion. Our whole kind of adrenal hormone system can get dysregulated. Our brain can get dysregulated because the tissues aren't really, kind of made, or they don't function optimally while they're being exposed to cortisol. It's kind of our emergency system. It's not meant to stay on all the time. And so when we have large amounts of stress or that system stays on for a long amount of time, that's when things can really get into this red zone here. We can get anxiety, panic, anger, breakdown, burnout, all of these types of symptoms. Here's a really good way to think of it. Stress is like our emergency system. It can save us once, right? And so a normal short-term stress response, we get the stressor and we bounce right back. But if we're exposed to repeated stress or chronic stress or really extreme stresses, or especially stress in childhood that is repeated or chronic or extreme, we kind of lose the ability to bounce back, right? It's like if you've ever jumped on a trampoline with another person, and sometimes you guys are in sync and it's great, you get this great double bounce, but sometimes you get out of sync and then you're not really bouncing at all, things just get uncoordinated. It's the same thing when we get overexposure to stressor, we get this, our whole system becomes uncoordinated and it becomes no longer able to really adapt very well. So we've got the natural state of things, the potential for conflict, resilience, the ability to overcome conflict, but then balance gets lost. And then we have our rescue mission, right? I had a professor when I was in undergraduate who said, if it's not a happy ending, it's not the ending of the story yet. I think that's a great lesson in life, equally applicable here, right? So when balance is lost, that's not a happy ending. So that's not the ending of the story. So we have the rescue mission. And the rescue mission is going to be analogous to kind of therapy, right? So our optimist Poppy and our realist Branch transform Bridget. So enter Princess Poppy, the optimistic leader. She's teaming up with Branch, the cautious realist, to save their friends. And I use ChatGPT or Microsoft Copilot here to give me a little extra content, so I'll read it. This collaboration represents the diversity of interventions that support the brain's recovery. These include neuroplasticity. If you don't know what that is, don't worry about it. We'll touch on it in a second. The brain's ability to adapt and rewire itself. Alongside psychedelics, key interventions include social connection, rest, mindfulness, therapy, nutrition, exercise. I maybe jumped the gun a little bit in giving you that. So let's just kind of keep moving forward. So we've got our rescue mission. Here's Bridget. She's with the trolls. The trolls kind of get this idea to model for her what healing can look like, really, what self-care can look like, right? So we see Poppy kind of modeling for Bridget, like, hey, I'm doing this self-care thing. I'm into colors and spunkiness. I can share that with you, right? Now, there are numerous analogies here. This movie is just rife with really fantastic analogies for healing. So the collaboration between Poppy and Branch really can represent the diversity of interventions, as I mentioned. Poppy also is very hopeful, very optimistic, very courageous. She really can be a representation of therapy and the things that we kind of really can help facilitate therapy. So I might even say that we need to be able to have healing and therapy. Poppy loves song and dance, so she also is just embodied in different healing modalities. We could add this as an analogy for psychedelic medicines. And then Branch, he's more of the realist, he's more pragmatic, he's more cautious. He kind of represents these kind of guardrails that we have in the psychedelic, in the science of psychedelics, in the psychedelic community, in the use of psychedelic medicines, right? He can also represent therapeutic integration. So we'll get into this much more in depth later in the class, but one way in which psychedelics act are that they can facilitate neurogenesis, so they can stimulate the birth of new brain cells, and they can enable brain cells to form new connections with one another. We call that kind of integration into the system. But if we're not intentional about that piece, there's also potential for psychedelics to just further ingrain cognitive patterns that already exist, right? So there is a real importance for caution. Branch can also represent the kind of underlying mechanisms or the neurobiology that are maybe a bit more set in their ways, right? And so we need that Poppy to come in and kind of break things up. So I mentioned neurogenesis. Now, here's kind of the brain equivalent of Poppy and Branch's rescue mission. In the brain, one of the main ways that psychedelics work are not, maybe one of several ways that psychedelics work, are that they can stimulate neurogenesis, which just means that they can stimulate the birth of new brain cells in the brain. So here we're seeing the human brain. We're seeing the cortex right here. If you don't know these terms, don't worry about it. We'll go over them. We're seeing the cortex. We're seeing the hippocampus. That's that brain region I mentioned is responsible for learning, memory, regulating stress responses, responding to stress responses, so mediating the way that stress impacts emotion or reward. And then this red ball here is the amygdala, which is that part that responds to aversive stimuli or threatful stimuli. These parts are highlighted, and then we're seeing the hypothalamus in there as well. These parts are highlighted because they're all known to have pretty robust neurogenesis that occurs throughout the lifespan. So including when we're adults, we are still actively producing new baby brain cells every day in these brain regions. And so just wanted to show the timeline here. We've got a neural stem cell. It goes through this process of proliferation. We get these neural progenitor cells. If this doesn't mean anything to you, don't worry about it. At about 3 days, the cell starts to differentiate into a neuroblast and then eventually into an immature neuron once it hits this survival stage, and then a mature neuron. Now, that's to some of you, that might be kind of cool. To some of you, that might be kind of overwhelming. That's totally fine. One point I want to make right up front here is that process takes at a minimum a month, 4 weeks. Better estimates are around 2 months, I would say. So we might start engaging in a healing modality, whether that's psychedelic therapies or meditation, mindfulness, exercise. It's going to take about a month for the neurobiological active mechanisms of that intervention to really take hold, right? So I also work as a health coach. I have this discussion with my clients that it might be about a month before you're really starting to feel changes kick in. Usually when you start engaging in therapy, you're going to feel a lot of benefits right away, right? Like you're taking an action for yourself. There's hope in that. You're connecting with someone. You might be getting things off your chest. Maybe not. But maybe. But usually it's going to take about, when you're getting into the deep work of trying to establish different behavioral patterns, different cognitive patterns, things like this, it's going to take about a month just for your brain to produce new brain cells that are mature and then integrate them into the system or form new systems or new connections that can really support new cognitive processes and new behavioral processes. So the rescue mission, pinning this back to the analogy, neurogenesis is kind of at the heart of it, and neuroplasticity as well. So the ability for these brain cells to form new connections with one another. And it kind of represents that innate healing that we all have inside of us, and we can use a variety of therapeutic mechanisms to tap into. So here's a little, you have watched it. If you haven't, this left. Oh no. I wanted to pause this real quick and just make the point that neuroplasticity, the ability for brain cells to form new connections with one another, does not require neurogenesis. Those are two separate things. And let me see if I can get us back started again. There's a variety of therapeutic mechanisms to tap into. So here's a little zoom in case you couldn't see it well. I meant to come into this when I was doing the other. But again, so neurogenesis is kind of the process by which new brain cells or new brain heroes are born, right? And then again, just showing the timeline there. This one I wanted to show just because it's a little bit more accurate here. It's telling us 42 days, so about a month and a half to get a new neuron to maturity, and then can take another month to kind of integrate into a new system. It doesn't always take that long, but it can. So this healing transformation, right, where we're undergoing change and our brain is actually changing to support that. We're getting the maturation of mature brain cells integrated into a new system. Now, one thing I want to say, don't get too caught up on these terms, radial glial cell, intermediate progenitor. Those are only there if you're curious. You do not need to know those. Not a big deal. So we also, in the movie Trolls, right, we see that Princess Bridget goes through this transformation. So Poppy kind of gives her this idea and they kind of run with it. And all of a sudden her hair is she's all getting glammed up, right? It's your typical glam transformation movie. So Optimus Poppy and Realist Branch help to transform Bridget. Let me see if there's anything else I want to add there. No, I think we're good there. So one point that I want to make here, and I kind of touched on the neurodiversity of neurogenic therapies. So neurogenesis, that process of new brain cells being born and growing, that is really central to a lot of healing and therapeutic processes. And there are a lot of different things, different types of therapies that can stimulate that. And I do not have the citation for this publication on here. Shame, shame, shame on me, but I'll put it in the comments of this PowerPoint slide. But this is from a PubMed indexed publication, I promise. And they're just showing different interventions that can stimulate neurogenesis. Now, this is a rat brain that we're looking at here, and this is the hippocampus right here. It's just a figure of a rat brain. And on the right, we're also seeing different factors that can decrease neurogenesis. So stress, glucocorticoids like cortisol, age, opiate exposure, excitatory amino acids. So if we get overexcitation, all of these things can actually decrease neurogenesis. So they can prevent or slow the process of new brain cells being born and growing and developing in the brain. And there are actually a lot of important reasons for that. And we'll get into that a little bit later. I think that is important to know because just as I said, stress gets a bad rap. We tend to think of it as a bad thing, but it does serve a purpose. And there's kind of an underlying message that I really want to convey in this class of like, we have this really, really beautiful neuroanatomy and neurophysiology that has developed and stayed with us and evolved through centuries, so long, right? And a lot of it has been really conserved and preserved. It hasn't changed much, which indicates to us that it's really important. And so when we're talking about using psychedelics or any intervention to really make changes, like lasting changes in the brain, in the body, in epigenetics, in the genome, I think it's really important that we have an understanding and a deep honoring and respect for the system that we're working in, right? For the landscape, for the body's innate ability to heal itself. Because there's also a very real risk with psychedelics, as there are with any interventions, of imposing too much onto the body, right? And we can impose risk when we're doing that. Even too much of a good thing can be a bad thing, right? So over here on the left, we're seeing interventions that can decrease neurogenesis, or over here on the right, over here on the left, we're seeing interventions that can increase neurogenesis. So this publication focused on exercise, enriched environments, and that can look like a lot of different ways to a lot of different people, maybe hanging art on the wall or going outside. Nature provides very, very enriched environments, just environments where there's a lot of stimuli, a lot of visual stimuli, a lot of sensory stimuli, and usually an element of safety and comfort as well. Hippocampal dependent learning. So we'll do, for the purposes right now, we'll just say learning anything. That can be learning a new skill. That can be learning how to do a handstand. That can be learning how to get to, a new location. So kind of map finding. Those are all different types of learning. Estrogen, just kind of by itself, can increase neurogenesis in adolescence. All individuals, men and female, go through this huge hormone burst that includes estrogen as well as testosterone. One big impact of that is it stimulates neurogenesis that gives us kind of all these new brain cells that enable us to just learn all, become kind of sponges in adolescence, learn all these different things. Meditation, mindfulness, nutrition, dance, play, novelty, like anything new, social interaction, and then of course psychedelic interventions, these can all stimulate this process of neurogenesis, of new brain cells being born and growing in the brain. It's pretty cool. So there's really a diversity of neurogenic therapies. Okay, so back to storytelling in science. We've covered the natural state of things, the happy force, the happy brain, the potential for conflict, the risk, the ability to overcome conflict and maintain harmony, our own innate resilience. We've talked about what happens when balance is lost, and we've talked about the rescue missions, therapy, healing, kind of targeting neurogenesis. And we kind of covered a little bit of the healing transformation, right? So that's when we get this neurogenesis, new brain cells get born, and then they develop. In this analogy, if we're going back to trolls, poppy and branch can almost be symbolic of therapy itself or of the rescue mission itself. Poppy really embodies song, dance, therapy, healing, color. Branch embodies these cautiousness elements. And then Princess Bridget is really where we see the healing transformation, right? So we kind of see her change. And then there's another element of integration that is so important in psychedelic science. So once these brain cells reach maturity, they then get integrated into new brain or neural networks. We talk a lot in psychedelic science, we'll talk in this class about therapeutic integration, right? And that can be as simple as learning a new tool or a new skill in therapy and applying it to your life, whether that's setting a boundary or approaching a new situation differently, or having radical acceptance, and just kind of practicing that and integrating it into your life. In psychedelic science, there's also a very large neurobiological period of integration. When we're using neurogenic psychedelics and we're stimulating new brain cells to be born or grow or to form new connections, which we call synaptogenesis. In brain science, we call it a synapse. When two brain cells kind of connect to one another and talk to one another, because there's a little tiny space, we call that a synapse. I'll get into that more later. When you're stimulating all of these processes, there's a period where things get integrated, where things are just really they're really like malleable and plastic. There's high neuroplasticity, right? And so in psychedelic science, we want to be really intentional about that period. But that is a period that can make this kind of healing transformation become lasting, right? And so, Bridget doesn't just dress up once, she kind of changes her lifestyle and she incorporates that into her lifestyle, right? And there's another layer of healing integration in the movie Trolls, because we see that Bridget's own self-healing spreads, right? So it goes from Bridget to the prince, and then pretty soon all of the Bergens are, in this technicolor, disco dancing, roller skating, super fun, healing energy and vibe, right? And so, again, the same is true of psychedelic actions in the brain. We might start with an intervention that could be psychedelics, again, that could be meditation, mindfulness, exercise, that are going to target this neurogenesis or neuroplasticity and many other things in the brain. We're just focusing on this one here. And that's going to give you like a one-time hit. And then, we have this integration process where those kind of healing mechanisms get integrated into the whole system, or those new brain cells get integrated into a new system. So we've got the healing transformation, the healing integration, and then of course we close it up with a bow. We've got the restoration back to harmony, to happiness, to the natural state, right? So we start once upon a time and we end and they lived happily ever after. Um, and in Trolls, I think it's really beautiful. There's this lesson of healing at the end, and this is a quote from the movie, where Bridget identifies happiness isn't something that you put inside you like a troll. Eating happy trolls is not the solution to finding happiness. It's already there, right? And that's another really big kind of lesson that I hope to share with you in this class is that, you know, I've been working in neuroscience for over 10 years, probably over 15 years at this point. And one of the biggest things that I've learned is almost any intervention that you might engage in to stimulate healing, you already have that innate ability inside of yourself, right? So psychedelics, for example, they can activate neurogenesis. We already have the ability to activate neurogenesis. And in fact, we're doing it all the time already, right? So I think that's a really important message, especially for folks in this class who might be interested in kind of going on to pursue, psychedelic work, psychedelic assisted therapy, plant medicine work, is these are tools to activate the innate healing that we already have inside ourselves and help restore us to our innate kind of way of being or harmony, right? So let's explore the brain's healing setup that's already there. All right. And now I'm going to switch back over to the PowerPoint, and I'm actually going to do the reverse of that. We're going to go into a little bit more of the stress piece, so the causative piece. Um, there's a few caveats that I would be remiss if I didn't offer first. One is that what I offered in that video was a scientific materialist perspective of the science of psychedelics, right? So I was really talking about what is going on in the physical brain. I think most folks who work in plant medicines and psychedelics, particularly anyone at Neuropa, though if you're from Neuropa, you've probably had me in class, know that there are other elements. Most folks who work in psychedelic medicines will say there are other elements that are at play when you're using plant medicines, So I also teach at the National University of Natural Medicine, where a lot of my students and colleagues, take a more kind of naturalistic approach to these plant medicines. They're really interested in how plant medicines can offer a way for our external environment to interact with our internal environment, and they will kind of talk about things like how can we, combine different plants together? How do indigenous cultures do that? I teach at Naropa. When I'm talking with my students and colleagues at Naropa about plant medicines, the conversations are often, they're often similar, but they're often very different. They're often talking about how the space is held and these kind of metaphysical elements that can come in, in the indigenous cultures that have used these medicines for ages and the lands that these plants come from, and to me, all of this kind of bodes the question of how might we endeavor to use these plant medicines that have been used for ages in a way that is in alignment with the way that they're used traditionally and also respectful and protective of the cultures and the lands that these medicines come from. My colleague Matthew Hicks presented on Friday evening to open up the summit for us. He actually started a study that interviews indigenous kind of shamans, medicine men, tribal men to collect their perspectives on this that I'm very grateful to be a part of. So it's a little plug. Um, there's a kind of also, if anyone is interested, I have a second lecture on humility in science that I think is really important when we begin to talk about these things that I think most researchers and folks who I know, we do what we do with a lot of humility, and most of my students who I teach at Naropa, when they come in, I'm teaching a research class. A lot of them have very negative perspectives on research. They'll highlight, kind of these more colonial aspects of research, which are very valid. They'll highlight, numerous gaps in our system, which is also very valid. Research, just like everything else, it's a man-made system and there are gaps in it. Um, they also highlight a perspective that, research and science thinks that, we can solve everything with research and science. And that, I would say, I've had amazing opportunities to meet my research heroes, almost all of them. And I don't think any of them would say, like, yeah, we've got it all figured out, or even, yeah, we're going to get it all figured out. I think part of being a researcher, I'm going to quote a friend, is getting a black belt in I don't know and humility, and I'll just say for myself, speaking to the metaphysical aspects as well, I'm a longtime student of Hawaiian shamanism and other indigenous practices, and I think that these really can go hand in hand. To me, neuroscience and research is a way, it's an art, it's a way to take a microscope to the most beautiful painting that there is. Okay, so that's my little homage. Let me let me keep going here. Um, there's also a couple of themes that were touched on in that video that we've kind of noticed throughout the summit. So the concept of neurogenesis, Neuroslapsis, who closed us out yesterday evening, talked a lot about nutritional interventions that can be used to support neurogenesis and brain-derived neurotrophic factor, which I kind of like to describe as like steroids for the brain, but it's endogenous, your body makes it. I have a video on that as well on our YouTube channel. The concept of innate healing, Erica Zelfand is going to talk quite a bit about in 2 hours. So hang tight. She'll also talk a little bit about excitatory amino acids, and then these concepts of connection, and a lot of other concepts that didn't touch on. The kind of concept as well of Poppy, the idealist, and oh, now I'm blanking on his name. The male troll character, the realist. I think Andy Novick is going to share with us a little bit his perspective on how antidepressants and psychedelic medicines can kind of be bridged, which I think will be really helpful. Okay, so that's all the plugs. Let me give just a very quick kind of recap of the stress system. So we've seen this slide already. Stress gets a bad rap. Nobody likes really to feel stressed out, but it is really helpful for us. If we have no stress, we often lack motivation, attention, focus. I'm happy to go into why that is. Too much stress, though, and this I think maybe tends to be the area where more folks in Western cultures live, can be equally harmful both to our physical health, our brain health, and our mental health, in large part because of the impact that overexposure to stress hormones has. And I think when we're talking about the stress system, it can be helpful to remember that this is our emergency system, primarily, when we're talking about big stressful events, right? This is our system that its purpose is to save us from dying and keep us alive. So it trumps all other systems in the body. The example that I usually like to give is if you're out foraging for berries and you hear a big growl and you look up and you see a hungry bear charging you. That's when your stress system is like, put me in, coach. This is what I'm here for, right? And it kicks all of these processes into place that in that context are extremely helpful, life-saving, critical for us to have, and make a lot of sense within that context. Now, within the past 200 to 300 years, socioculturally, we have evolved extremely rapidly. I mean, we used to have an ice block press at my house from, my grandfather's father who chopped ice for a living, for people's ice, kind of freezers, what they had then. And now we can order anything we want on an app from anywhere in the world. And I would argue that our bodies haven't necessarily adapted so quickly. Genetics are the rate of genetic adaptation is limited by the rate of our lifespan. Of course, we have epigenetics that, can enable that to happen a little more quickly. But I think there's a lot of situations where our stress system gets enacted and the things that it does that are extremely helpful in the context of we're eye to eye with an angry bear and we need to be running for a long period of time and we need to be focusing on nothing else. Aren't so helpful when our stress system might get kicked on in the middle of work and we can't run and we need to be focusing on our work, right? I think that's important to mention because I think a lot of times when stress can kind of wreak havoc on the body, those who experience it, we can think that we're broken. We can think that there's something wrong with us. And I don't think that's true. I think that we just have a little bit of a mismatch right now in the environment that we live in and our biology, that has evolved over, many, 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 many years. And I'll just, kind of stop there. So stress, it gets a bad rap, but it is critically important. We've seen this slide as well, so I think the trampoline analogy is a good one since our stress system generally is our emergency system. One kind of hit and we can bounce back, but if we have too many, that's when our stress system can start to become uncoordinated and dysregulated. Now, I want to make a point that there's different types of stressors here. So, acute stress, which would be like a one hit, one stress exposure experience. Sorry, this figure is really small. Um, that's one type of stress, we'll just say. So if we have one exposure to stress, that's going to be acute, it's going to be transient, meaning it goes on off, might be kind of a just enough. Sometimes stress can be very good, right? So in these encounters, we might, have an exam that we need to study for. We're going to get a little bit stressed out. That's going to turn on our stress system. We already kind of talked about that HPA axis. That's going to increase levels of glucocorticoids, which is just a fancy name for stress hormones in our brain. These are going to be things like corticotropin releasing hormone, CRH, which is also called corticotropin releasing factor, CRF, ACTH, and then cortisol in humans, or corticosterone, you'll hear it called in rodents. We also have some neurotransmitter systems that are turned on. These are going to largely be catecholamines, which is just a class of neurotransmitters. So these are going to be things like norepinephrine or epinephrine, which is also called noradrenaline or adrenaline, acetylcholine, 5-HT is serotonin, and then DA, dopamine. We're going to have these behavioral responses to stress that are largely very beneficial. So we get kind of increased arousal and vigilance at the neurochemical level. That's largely supported by changes in corticotropin releasing hormone, norepinephrine, and acetylcholine. We're going to have a better ability to process external cues. And then we're going to have enhanced motivation, concentration, and attention. It's going to be largely from dopamine increases in the nucleus accumbens, and as well as increases in acetylcholine, and then cortisol kind of drives that system. And then alterations in our reward responses. And in small doses, stress can actually increase, as I mentioned, dopamine output in the nucleus accumbens. So that's going to actually increase our reward response and our reward value. So that enables us to assign a higher value to whatever it is that we might feel stressed out about. Let's say it's an exam. And that's going to enable us to prioritize studying for that exam over, maybe going out to dinner with our friends or other things. So this system is extremely adaptive. When we get really extreme amounts of stress, so now we're not talking an exam, we're maybe talking, what we might think of conventional adverse childhood experiences. So abuse, bullying, things like this. We're going to get very high levels of stress hormones, and those tend to be not so great for the brain. So we're going to see diminished decision making. We're going to see increases in impulsivity, reduction in goal-oriented behavior. So we often become less able to concentrate, focus, because we'll see alterations in dopamine output in the nucleus accumbens. That's going to diminish our ability to focus on long-term goals. We're going to see habit formation occurring, impulsive habit formation, and then alterations in reward learning. So increased positive reinforcement. So we're going to have stronger reinforcement around things that feel good or have positive responses, and reduced negative enforcement. So we're going to start to respond less to things that have negative impacts. And let me rephrase that. So negative reinforcement is when we engage in something, it has a negative response in us, and so we learn to avoid it. And it's that avoidant part that is diminished. So we don't avoid as much things that are negative. This is kind of a setup for drug addiction, or many habits that can be formed under stress, where we might return to a behavior or an action or a stimuli source that feels positive to us in the short term. Because remember, we're talking also up here about impulsivity. We have less focus on our long-term goals, and we respond less to negativity. So that's kind of a pretty classic setup for drug abuse, or kind of what we call addiction. And then some other types of stressors that we can have chronic stress, right? So where we have repeated stress exposure, or stress that occurs for long periods of time. Here we're going to see alterations in regulation of that HPA axis. And I think I have I have a couple of slides coming up. So we'll just hang tight on this one for a minute. But remember we talked about that HPA axis that coordinates hormone responses to stress, has kind of a thermostat, right? So the hippocampus, when cortisol or stress hormones act on it, it turns off that system. And we see that start to get dysregulated when we have overexposure to stress. And part of that is because the brain cells in the hippocampus start to die, right? And so as the brain cells die, the hippocampus becomes weaker and it becomes less able to turn off our stress system. Similar mechanisms happen, other mechanisms in the amygdala. So what we usually tend to get is the stress system becomes sluggish. It becomes slow to turn on and slow to turn off. And the stress response, so the amount of hormones that are secreted into our body, tend to become prolonged and stand longer. And then all of this together can lead to a large variety of psychiatric disorders: post-traumatic stress disorder, addiction, obsessive compulsive disorder, depression, anxiety. I'm almost at 9:50, so I'm just going to really quickly touch on a few of the brain mechanisms. So we've seen this slide before. This is the HPA axis. So when, let's say we see a visually aversive stimuli, a spider in the example I gave, it's going to activate the amygdala. There's going to be some other processes in the brain that are activated, the hypothalamus. That's ultimately going to induce secretion of corticotropin releasing factor, which is a stress hormone in its own right. It's going to stimulate the pituitary gland, release ACTH, another stress hormone, stimulate the adrenal cortex, stimulate cortisol to be released in the body. And then that cortisol, because it's lipophilic, it's going to act on different brain tissues. So we've already gone over this. We've talked a little bit about the amygdala and the hippocampus. I'm just going to give one more layer of kind of the effects here. So some of the regions that have a high amount of stress hormone receptors, meaning it's going to make these regions really sensitive to stress, are what we might call limbic regions. So I'm going to just go over a few of these in the couple of minutes that we have left. So the amygdala, here's where we're seeing the amygdala in a human brain, tends to be associated with fear, anxiety, social behaviors like aggression, as well as copulation, mating, and PTSD. The neurotransmitters that kind of drive that system are corticotropin releasing hormone, serotonin, norepinephrine, and then also inflammation in the amygdala can drive these behaviors. The amygdala activates the HPA axis, turns on our stress system, as we've kind of went over. And when we get overexposure to stress, so stress in the amygdala is going to increase function of one part of the amygdala, the basolateral amygdala, and reduce function in another part of the amygdala, the medial amygdala. Overall, this is going to result in enhanced anxiety behaviors, fear behaviors, and then behaviors akin to PTSD. The amygdala is very highly associated with storing aversive memory and responding to threat, right? So when this part of the brain gets dysregulated, that's when we can kind of have more of these behaviors. The hippocampus, which is a part of the brain that I studied, has kind of two parts to it, a dorsal or a back, if you think of like a dorsal fin, a back side of the hippocampus, and then a ventral. So if you think of like your stomach as being your heater or your vent, kind of the anterior part of the hippocampus. So it's got these two parts to it. The dorsal hippocampus tends to be associated with learning and memory. So episodic memory, just kind of remembering routines, daily events. That's going to be largely kind of supported by dopamine and norepinephrine. Spatial memory, so map finding, navigating space, it's going to be largely mediated by dopamine and norepinephrine, again in the dorsal hippocampus. And then the ventral hippocampus, this kind of underbelly side right here, which is the part that I studied, is associated with regulating stress responses. That's the part that turns off the HPA axis and the stress system, largely mediated by glutamate and cortisol. And then it's largely involved in mood regulation and essentially enables stress to impact mood through its connections with all these other brain regions. It's going to be through dopamine, cortisol, and serotonin primarily. The ventral hippocampus can help turn off the stress system, terminate stress responses. When this part of the brain gets overexposed to cortisol, we're going to see the brain cells die. That's where we're going to see cognitive deficits, impairments in memory. This can lead to Alzheimer's disease. We're going to see increased depression. So brain cell death in the hippocampus is correlated with in humans and causes in rodents depression and depressive-like behavior. And when you restore brain cell life through neurogenesis, that correlates with reversal of depression in humans and causes reversal of depressive behaviors in rodents. And this is thought to be through neurogenesis. This is also thought to be the mechanism that antidepressants work through. The serotonin system is largely interactive with BDNF and neurogenesis. So antidepressants that's one mechanism by which antidepressants are thought to be able to improve depression. This is one mechanism by which psychedelic interventions can also improve depression. Same with exercise, good nutrition, things like this. And when we start to see this part of the brain become impaired, we start to see impairments in stress regulation and stress coping. So this is just kind of a little bit more on the two parts of the hippocampus. The dorsal part of the hippocampus associated with spatial learning and memory. It's also associated with exteroceptive stimuli, which is just a fancy word for processing information that comes in from our external environment, from our external facing senses and receptors, so sight, smell, touch, sound, things like that. Highly associated with depression and mood because of cell death and neurogenesis. And then we see high levels of neurogenesis and neuroplasticity in the dorsal hippocampus, which makes sense, associated with learning and memory. And then the ventral hippocampus is that part of the hippocampus that mediates the stress system. And I've already kind of mentioned that, but it's also associated with interoceptive processing. So processing internal stimuli about our internal environment. So this would be information from our different receptors and senses inside our body. So baroreceptors, nociceptors, tempororeceptors, thermoreceptors. So receptors that give us information about our body's internal temperature, pressure, when things are stretching, pain, things like this, chemical changes, stress hormones, things like that. So the hippocampus is also a region that is highly involved in integrating external information and internal information. I'm going to go 2 more minutes and then I'll stop. I'll leave 5 minutes for questions. And I saw Corey log on a little bit ago. So I imagine that's maybe why most people are here. So we'll kind of get into the good juicies in a little bit. And then just a final point that the ventral hippocampus, as well as the dorsal, connect to a large variety of other brain regions. One of those is going to be the nucleus accumbens, shown right here. So we're seeing the ventral hippocampus projects to the nucleus accumbens, which you'll also hear it's part of the ventral striatum. This is associated with reward value, reward sensitivity, reward reinforcement, as well as motivation, attention, and focus. So this is actually what my dissertation work directly explored. The ventral hippocampus is one mechanism by which stress can produce alterations in the way that we value different things going on in our life, our ability to regulate our reward responses, how sensitive we are to rewards, and how reinforced we are by rewards. So stress can really kind of prime a system to become very sensitive to rewards, more vulnerable for developing addictive patterns, whether that's in eating, eating foods that maybe don't align with our long-term goals, but we get an effect. Nancy Cayley talked about we get an effect from them when we eat them that is more immediate. And then because of the impacts on motivation, attention, and focus, we tend to see that shift to things that are more immediate when stress is on board. Which again, if you think back to the evolutionary nature of stress, it's there to save us from dying. So in the context of stress, from our body standpoint, we need to be paying attention to the immediate, right? It doesn't matter what we're going to do tomorrow if we die today. So, in that context, this all makes a lot of sense. But again, our culture has evolved so rapidly, over the past 200 years, and our brains and bodies haven't necessarily adapted with the culture. I'll put it that way. So that's a ventral striatum or the nucleus accumbens. It's going to be the striatum here we're seeing again involved in motor responses as well as reward responses. I'm just actually going to scoot forward here a little bit. We're getting ready to land the plane. Oh, I thought I had a different slide here, but apparently I do not. Um, so there's kind of two parts to, oh yeah, we do. Okay, I'll just keep going here. Um, so the striatum largely it's going to be involved in motor and action planning, so planning movement, as well as decision making, motivation, reward value. Salience is a kind of, you can think of it as value, reinforcement, habit looping. So this is where we start to see kind of rumination in response to stress, as well as habit learning. Um, and then when little amounts of stress are on board, we see increased motivation, increased reward value, increase in habitual behaviors, and then and increase also in drug taking, food taking, and then when we start to get a lot of stress on board, that's when we can start to see a lot more dysregulation. So we can actually see reductions in motivation that can align a lot with depression or anhedonia, um, you know, alter dysregulation of reward value, things like this. And then that habit behaviors can really get solidified. Okay, and then the last brain region I'll talk about here is the prefrontal cortex. So the prefrontal cortex is the very front of the brain, as we're seeing right here. It's actually the last part of the brain to develop. The brain kind of develops in a bottom up and center out fashion. So the prefrontal cortex, which is involved in executive function, behavioral control, it actually doesn't fully develop until young adulthood, so around age 27. So, when you think of adolescents and we tend to think like, oh, they're unruly, they don't have good ability to regulate themselves, they don't think ahead. Often that's because their prefrontal cortex is still developing. They don't actually have the makeup to do that. Folks who work in the psychology, parenting, child psychology space, I've heard often say, one of the primary roles of a parent is to be the prefrontal cortex for your child until they can develop one on their own. So it's going to be involved in decision making, working memory, and then self-regulation. The prefrontal cortex has inhibitory projections onto the amygdala, the hippocampus, the striatum, all of these limbic regions that we just went over. And it enables us to kind of suppress those so that we can focus on long-term goals when stress is not, is less on board. It also helps terminate stress responses. And then when we get a lot of stress, that starts to impair the prefrontal cortex. That's when we start to see deficiencies in executive function, increases in impulsivity, cognitive rigidity, habit forming. We start to rely more on information, quote unquote, from our limbic brain regions. And then this can also walk us down a path of substance use and addiction, as well as ADD, ADHD, other mental health issues. This slide was from a presentation I gave on stress and addiction. So that's the limbic system. And I'll just give a very quick, so we've got the amygdala, fear, anxiety, stress, the hippocampus, learning and memory, regulating stress responses, and then the ventral striatum. It's got these two parts, one associated with movement, one associated with reward. And here it is all together. And do I have an ending slide here? I believe I do. Yeah. So, we've got 1 minute left. I'll maybe, if anyone has any burning questions, we'll take them while I'm kind of just queuing up things for Corey. And if not, we can just move right ahead.